So, and just like before, I will try and cut out the stuff that's less important. So we're looking at cell size limitations. Cells can only get so big. In fact, I'm gonna scratch out of that. So you will want to write this down, actually. So there will be a little bit more notes, but this will overall be a time save. And I will show you a visual thing of this right after. So with cell sizes, cells are really small, right? They're tiny. They're smaller than the tip of a needle. So when we have a cell, we have a surface area, and that's covered by that plasma membrane. This is not new to us. And then the stuff inside gets taken up by the inner contents of the cell. So basically, when we have cells with a higher surface area to volume ratio, and I will explain what that means, they can more easily sustain themselves. Again, cells with a higher surface area to volume ratios will better sustain themselves, and I will show you that in a second. Okay, so what does this mean? A surface area to volume ratio can sustain themselves more easily. So what I have here is I have 3 different cells. So we have a nucleus here, and around it is different sized cells. So if we're saying that, hey, a higher surface area to volume ratio means that cells can more easily sustain themselves, this means that we want a lot of surface area for not a lot of volume. We don't want a lot of stuff inside. We want a lot of stuff on the outside. It means that it's more compact, and you're better able to it's better able to regulate itself. So all the nutrients pass through this membrane, right? And all the waste exit out of it. So if a cell is too big, it can't communicate effectively. It can't control itself properly. If it's too small, there's nowhere to fit the nutrients. So you kind of want somewhere that's a little bit in between. Again, if you think about a shower drain here, if you have a really big drain, this is never going to flood, right? It can drain more than what the shower can spit out. It's that you're not going to flood unless you block them. So if the drain's big, it's not going to flood, and it's not going to overflow even if you have a lot of water coming in. But if the drain is small, it's going to flood, it's going to overflow, it's going to clog up. It's not going to work as intended. So so where we're kind of going with this is when we're looking at cells, again, with this ratio here, those smaller cells can better transport the substances easier. They can take in waste, so they can spit it out waste, and they can take in nutrients better. But that diffusion that happens, diffusion is when stuff transports across that membrane, right? One of the first things we learned. The bigger the cell, the more inefficient that comes. As well, those skeletons that some cells have, those cell walls that some plants have, they're weaker the bigger you get. So generally speaking, smaller cells are more efficient. The smaller cells are more efficient. As a general rule of thumb, there are exceptions. Okay, so and again, just kind of going back to this thing here, smaller cells are usually better just because, again, the surface area to volume. What does this mean is this one over here has way too much surface area for just how much you can fit in it. It becomes inefficient to sustain itself. So once we reach a size, cells grow, right? Cells grow, they mature just like us. The thing is though, once it reaches a certain size, again, if this cell in the middle is to go to this big one here, we just established that it shouldn't do that. That's not good for its survival. So what it's going to do is instead of growing, it is going to stop growing or it's going to divide. So one cell will become two cells. So this growing and cycle process is called the cell cycle, and that's primarily what we're focused on today. And again, every time it goes through one cycle, it turns into two cells. And so we got three main phases of it. We're gonna break down all three. Okay, so again, we have three different stages of the cell cycle. And what is the cell cycle? The cell cycle is one cell turning into two. This is the top part is really the fancy way of saying it, right? A cell either has to stop growing or it has to divide. It can't keep growing. It'll kill itself if it does. So with this cell cycle, it goes through three phases. And we're going to learn about all three of these. But so we have interphase. Where's my pen? Oops. So we have interphase. This is when the cell grows, it replicates its DNA. We have mitosis. This is when it actually divides. And then we have cytokinesis, which is when the cytoplasm divides. So all of this is the cell dividing, one cell into two cells. So this diagram, you do not need to draw. I have this printed off for you guys that will be given to you today. But this is what the cell cycle is. So again, so with the cell cycle here, all of this stuff here is that interphase. So that's all that interphase. If you remember, interphase, mitosis, cytokinesis, that's what the cell cycle is. There's three primary things, but the interphase is so big, it gets split up a little bit. So interphase has G1, S, and G2. During this first part here, G1, which is most of it, this is when the cell's growing. This is just the cell existed. So all G1 is it's growing. And it's in the name. It's G. The G stands for growing. There are some exceptions to this. Nerve cells, brain cells. Brain damage is permanent, right? If you damage a brain cell, it cannot replicate itself. If you damage a nerve cell, it can't replicate itself. Once those nerves are gone, that's it. If anyone here's ever had a really bad burn or a really bad scrape, maybe you were biking like you fell, you might have lost some feeling in some parts. It doesn't come back. Okay. So there's three phases, right? The G1 is going to be our growing. S is going to be our splitting. And I'll talk about G2 shortly after. But we like the acronym stuff. If you remember GSG, growing, splitting, and what the third thing is, it'll be easier to remember. So again, during G1 here, it's just the cell's existent. That's it. The cell exists and it does its normal stuff. I'm not too sure. So during our S phase, S is what I'm going to recommend is it stands for split, technically. It's preparing for cell division. It's preparing to split. This is not when it actually splits. In your phase is all about preparing itself. You know, when a bear goes into hibernation, what does it do? It eats like crap. It's preparing itself for the winter. But during the S phase, it copies its DNA and it creates the split it. Think about if I need to give you files on a USB, I would copy it from my computer to my USB before I give it to you. So we're going to eventually look at chromosomes, and this is where we're going to spend a lot of time looking at. The next part here, the rest of the course is genetics. How are traits passed on? What are chromosomes, etc. You don't really need to know this part. Okay, so again, during S, we are synthesis. It is getting ready for cell splitting, cell division. And then finally, we have the G2 phase. So this is when we prepare for the cell. This is when the cell prepares for the division of its nucleus. So G1 it is copying it. G2 is when it actually prepares the rest of the cell. Again, G1, it's when the cell just exists. It's just doing its stuff. It's growing. S is when it's now grown to its peak. It needs to now start to get ready to divide. So it starts to copy its important stuff. It's putting its information on a USB. The G2 part of it is now it's preparing the rest of the cell to actually divide. It's going to gather up energy. It's going to rearrange itself, etc. Okay, so those are our three phases. Here, we'll do this quick. So we're going to learn more about mitosis next section. In fact, we'll learn about that later this afternoon. But basically, during this process, which we will learn more about, this is just a little part. It's not a lot, right? During mitosis, this is when it actually divides and separates. This is when you actually get the 1 into 2. The whole thing before this, most of this is just getting ready. So mitosis is when it actually splits. At cytokinesis, this is when it divides into the 2 daughter cells. And we will look at the difference between these two. Because right now you're probably looking at this and you're thinking, well, isn't this the same thing? It won't be. And it will make sense, I promise.
Basically, isn't that saying nuclear material divides and separates into all sides of the cell? It's like X, Y. You're gonna get your dad's side and your mom's side.
Oh, we're gonna learn about genetics soon. That is in here. We'll learn about that, how that works. Yeah. And sorry, that's nucleus, not nuclear. That's my spelling mistake. So here's one thing that's not on here that I'm going to recommend you guys put in. How long does this take? So if we think about this, when you are conceived, you're a single cell, 1. When you're born, there are, I actually don't know the full amount, billions or trillions of cells in you. How long does it take from one cell to double and then double again and then double again and then double again to reach, a trillion? How long does that take? For humans, it's 9 months, right? Humans, it's 9 months for a baby to be born. But how long does the cell cycle take? How long does it take for that first cell to turn into 2 cells? The cell cycle lasts 12 to 24 hours. So it divides once or twice a day. So yeah, only once or twice a day, yeah. Okay. So last minute notes for a bit. Let's take a break after this. Not an actual break, but we'll change what we're doing. So when we're looking at prokaryotic cell division, these eukaryotic cells go through the cell cycle. So everything we just talked about is about those eukaryotic cells, right? Those complex cells. The prokaryotic cells, those are the really simple ones, the really small ones, go through something called binary fission. That's not really that important to us today. The big thing that we really care about for this entire section is the cell cycle, mitosis and stuff. But whenever we talk about that, it's just important to note, we're always talking about eukaryotic cells. Prokaryotic cells are just so simple, we don't really consider it that much. Okay, so I think everyone has that name. Okay, so that's going to be it for the first part here. We're gonna creep into section 2 later. That'll be probably after the break. So what I'm going to have you guys do is I don't want you guys to have to read the full textbook for this. We've done a lot of reading, and it might not even be that effective. So instead, what we're going to do is I'm going to give you guys a chance to kind of glance through it. Specifically, you were given two handouts. I'll quickly talk about those. One of these is for marks. These ones here, these quick checks and those study guides are going to be for actual marks that you'll get some time to answer. The other one, though, and for those of you online, I'll show you where this is some of those notes. Again, I've tried to reduce the amount of notes that you do have to take with this. All I want you guys to do with this is I want you to go into the text. I'll make that red, I'll make that bigger. I'm going to want you guys to go into the textbook. And I'm just going to want you to fill out these blank sections. Right? So just some of these blank sections within here. You can see them here as best you can. So anything that you'll see here, anything that's blank, I want you guys to try and fill out. It's not going to take that much time. You don't need to fully read the textbook. You can glance ahead to certain sections. It's just the main takeaways I want. So you guys are going to get about, I'm going to say like 20 to 25 minutes to 1, do these essentials, and 2, to try out the quick check for chapter 1. Now, for those of you online, you can find that here in the chapter 9 section. There's just these 2 PDF documents. So there are 2 documents. I need to print you one off. I swear I did, and I've lost it or something. Did I give you one extra? Yeah. Oh, you want to pass that to her, please? Perfect. I was like, I knew I printed 4. So I'll give you guys here about 25-ish minutes. You're going to want to grab the textbook. You're going to go through it and do both of those for section 1. Both of those. And if you're not sure, that's fine. Ask, because we're trying to power through this part for the sake of time, so we can also get to the good stuff. So now hopefully you've had time to do that. I just want to go over a couple of the questions with this. So I got 4 questions here. Oh, I'm looking at the wrong one. I got 4 questions here just about this. So I'll give you a minute just to copy those down. In terms of how much space, probably just 2 lines between each question would be long. 2, 2, 2, 2. Going through these questions here. Here we go. So why is cell size limited? You cannot have an infinitely large cell. It's because when we transport those nutrients and wastes across that plasma membrane and that cell control, it becomes difficult when cell sizes increases. Basically, you can't have too big of surface area. This is a really long-winded way of saying that. In fact, I'm just gonna do this. So this bottom stuff is really the important part. Okay. So the next part here is, what are the primary stages of the cell cycle? Interphase, mitosis, and cytokinesis, right? So remember, in interphase, there's too many stuff open. So in interphase, the cell is just existing and growing, right? In mitosis, it's splitting the nucleus, not the cell, the nucleus, still one cell. And in cytokinesis, it's splitting the cell. There's now two of them. Okay, next one here is what happens during the S phase of interphase. And if you recall, we have G1, then it goes to S phase, and then it goes to G2 phase. And the DNA gets replicated during it, essentially. For the fourth question here, I'm not gonna, like I said, this is already in your notes there from that handout, so you get to keep that. The next one is, well, what happens if we have a really large cell? And this managed to divide despite the fact that it had grown to a really large size. So if I have a really, really big cell, what's going to happen to it? If I decide that, you know what, it's time this thing splits. So it's grown, it's going to split up. So what's it going to do? It's going to spread its material out like this. And now what's happened? Yeah, let's make that. So what's happened here is we know that a really big cell, one that's too big, is inefficient. It's more likely to die. Now I have two smaller cells. That is a better ratio. Those two now cells have a much higher probability of surviving. Okay, and so that's it for section. That's it for section 2.1. Yeah. So what we're going to do now is we are going to carry on forward here to section 3.2, but we're not going to immediately do with notes. You guys have done a lot. Instead, what we're going to be able to do is give you guys a chance to read section 3.2. You'll only need to read it. You're going to try and work on the essentials, work on the quick checks for 3.1 and 3.2. Tomorrow, we'll officially go over it because this is when we start to get into stuff where it's really going to get a little bit more interesting. We're going to start seeing how cells actually divide and stuff. So that's what we're doing today. So you guys get a chance to read section 3.2. And if you haven't yet, it's a good time to be able to finish up your stuff for section 3.1. So my hope is tomorrow we will finish all of this chapter, is my hope. But by Wednesday, for sure, we will.
Do we have to have these by tomorrow?
No, you won't have to have those done by tomorrow. No.